Second practical is going to be a very, very similar format to the first one. You remember the first one? Sort of, it seems like forever ago, right? But it's going to have the same basic setup. It's going to have all the section specimens are up here. So it's going to be, it won't be in order necessarily where you go respiratory system, digestive system, right? Respiratory, digestive, urinary, reproductive. So it won't necessarily be in order, but it'll be first question over here, rotate through, last question will be over here somewhere. So 100 questions. And we tend to concentrate all the dissection specimens in one place because I end up having to do all those. So I get to do all of it, yeah. So we'll do, there will be 7 or 8, something like that, specimens, which could include rats and pigs, and the kidneys, right? We had those disembodied kidneys on the that were available to you. Which one? Wrong class. You're getting mixed up. She's doing A&P1 at the same time. So, yeah, and there'll be pin structures. So, the things that you need to be responsible for, say in the respiratory system, are those things that are starred, right? Those little asterisks, whatever they call them. Some are less likely to be pointed out than others. So I'm on page 105 in the lab book, which is that table page. Now, I'm just going to tell you, I'm not going to put a pin in the pharynx of the thing and ask you to determine where a nasopharynx or laryngopharynx is. I'm not going to do it. And I doubt your teacher's going to do that either, right? So it's going to be the structures. Whenever I suggest how to study, I always study the big things first, especially in the practical. Study the big structures first. So you better know what the lungs are, right? And that is something that you can mess up. Do you remember those quizzes? I think we had a quiz on respiratory system. I had a student. I had to pin it in the lungs, and somebody said liver. Right? I mean, it can happen. It's a simple thing. So when you take this test, slow down. You got plenty of time. It's an hour and 15 minutes, which may not seem like a ton of time. 75 minutes for 100 questions. But if you just approach it in a slow, sort of methodical way, you're going to move a lot quicker, right? Slow is careful, and careful is fast. Thanks. And if anybody didn't sign this, sign it on your way out. So start with the big things, work your way down to the small things. So if you see the trachea, right, you'll know that that's connected to the lungs. Kind of go through these things in a careful, thoughtful process. So maybe, maybe like out of 100 questions, maybe 30 of them or 25 of them or something will be on dissected specimens. Then we'll move here into a combination of maybe some models and some microscopes. So we'll have a heavy dose of slide issues. And I'll put some of those things up on the board. I'll show you guys some slides towards the end, just to test, kind of a test prep. But when you come up to any of these questions, my students might know this because I probably told them, but what's the first thing you do when you walk up to a question and a specimen? Read the question, right? Don't look in the microscope. Don't look at the model. The model's going to be a big thing that has a sticker on it or has some stickers on it. Don't start looking at those. You're going to probably cause more mistakes than you're saving yourself time. You won't save any time. So go read the first question. It says, list identify the structure at pin whatever, Z or whatever, right? Then go find pin Z. Make sure you answer the question correctly. And this is something that's hard to make yourself do. It's hard to make yourself remember that it's asking for a function or remember that it's asking for a structure. But there's going to be structure and function or deduction type questions. Don't answer the wrong question. Nobody can give you the right answer, give you the correct answer to the wrong question. If you answer the thing what it is and it didn't ask you that, it asked you what it does, sorry. You probably knew what it did, you just didn't answer it right. And then there will be a bunch of models, and we've got a plethora of models. We have those, most of them are put away, the respiratory system models, the urinary system models, we've got kidney models, we've got entire digestive system models. And they're all kind of over there in those cabinets. You've all seen them. And again, for those, a lot of replicas of those, similar ones are in the Science Learning Center. I'm assuming that most of you have been over there regular enough. If you've been over there this week, it's probably been busier than usual. And if you go over there next week, it's going to be way busier than usual, right? So if you can squeeze in any remaining Science Learning Center trips this week, do it, because it's going to be A&P1 and A&P2 fighting for space, right? Crowded. All right, so same type of questions, different type of material. You guys won't be surprised by the format. All right. So let's go over some typical things, some things that maybe, I can't guarantee these are going to be on the test, but I can tell you that I've seen a lot of these things on the test, right? So some real areas to focus on. I'm looking at the respiratory system first, okay? Besides for the parts, those volumes. Remember those the graph of the respiratory volumes, or at least the terms. So you've got this sort of thing where tidal volume, inspiratory reserve volume, expiratory reserve. Oops, my lungs. I've drained them. Expiratory reserve volume. Then this would be total lung capacity, which goes all the way up to here. And there's a couple others, but stick to the main volumes first: tidal volume, inspiratory reserve, expiratory reserve, and vital capacity. Remember, the capacities are sums of volumes. So they'll probably stick to those winners. Let me just roll through the chapters, and then I'll find slides and do them, and maybe a couple of models if we have time. This one seems minor, but if you miss one question, if you could miss one question on this test, if you could go in knowing you'd miss a question only, you'd take it, right? For sure. But then, when does one question become 2 questions, become 5 questions, become 10 questions? At what point is it not acceptable? And usually that's somewhere between 90 and 80, right? You're like, oh, I really don't want to get a 60 on this. Nobody really wants to get a 60. So when you miss one question, you might say, chalk it up. But that one question can happen a bunch of times. So some of the minor stuff, you definitely want to be aware of. The muscles that control respiration. Not just identifying them. Some of them, like on the rat, you can see the diaphragm. That's about it. But there's intercostal muscles, external and internal. Which one of those is responsible for you taking a breath in? External, right? And then when you breathe out normally, when you don't breathe out forced, you don't use any muscles, you just relax, right? But if you want to breathe out forcefully, which muscles? Internal intercostals, and what? And your abdominals, right? Your rectus abdominis. Because if you're really blowing out a pile of candles, you're going to crunch that gut too. So you can involve those guys, and they're listed. Let me show you guys something that students have. I'll do some, not just review. I'm going to show you something. So here is my nose or mouth or whatever. It branches into this is my respiratory system, and I'm going to be real simple here. So those are my lungs, right? Trachea, larynx, and then right here, I'm going to draw these in like these are the ribs. So this is my outer body wall. This is my thoracic wall. So there's a layer of tissue on the inside of the body wall, and there's a layer of tissue on the outside of the lung, right? What's the name? They're both called serous layers. That's the ROUS. What's the serous layer that sticks to the organ? The visceral layer, right? And the serous layer that sticks to the ribs is the parietal layer. And they're named by location. So this is the visceral pleura and parietal pleura. Down here in the gut, it would be the visceral peritoneum and the parietal peritoneum. So what is this chamber in here called? It is the pleural cavity. So the pleural cavity is the container that holds the lungs, right? It's part of this thoracic cavity in general, but individually each is held by a pleural cavity. This person right here, this is the atmospheric pressure out here, whatever that is. 760 millimeters of mercury, or 1 atmosphere, or whatever, how many bars, whatever, doesn't matter. So this air, if I took a BB and sniffed it, it would go, and it would just plinko its way down here until it got all the way into these little sacs, right? Called alveoli or alveolar sacs. But what that means is that this air that's in this space is continuous with the air that's in this space. So if you're not closing off your air pipe, atmospheric pressure is the same as this pressure. What's this cavity or this pressure called? Intrapulmonary or intraalveolar. Right? So intrapulmonary, intraalveolar pressure, this is intrapleural pressure. Pleural. Pleural cavity, intrapleural pressure. Pulmonary lungs, intrapulmonary, intraalveolar. This pressure, let's say this one's 760, this one's 760, this one's less. It's like 4 millimeters of mercury less or whatever, but it's less. Why is this one less? It's so that the lungs don't collapse, so it sucks the lungs to the walls. Right? So kind of know that just knowledge of that. Don't try and memorize the numbers necessarily, but just know that this pressure is lower, and know the names, and that tells you a lot about how respiration works. And there will be a couple of questions on that. So, for instance, we have our diaphragm that goes up in here in a bell, right? And so when I want to take a breath, it's going to flatten out like this. It's going to go. What it does is and that tells me about that whole Boyle's Law thing that we talked about, that you went over, right? Boyle's Law, if I want to increase the volume, it decreases the pressure. If I decrease the pressure of this thing, that decreases this pressure, which then decreases this pressure, which then blows air down my nose. And if I want to go the other way, decrease volume, increase pressure, air blows out. So anyway, try to understand the stuff as got like some bubble voice here. We get like we go, oh, start talking like a Muppet. Try to understand the stuff as a whole picture rather than try to memorize line items. If you're going to have to call certain things out of your brain as line items, but memorizing them as such is not going to help you recall them on the spot. You want to understand how the whole thing works together. All right, urinary system. What do we got inside of the animal? What can we see in the rat and or pig? Kidneys, right? Ureter. Urinary bladder, and that's probably about it, right? You might get to see. Doubt you can see the urethra. It's scarred there, but you can't identify it, right? So there's only 3 things kind of in the urinary system, and they're all located in certain spots. When we get to reproductive system, those tubes that lead in the female's reproductive system look kind of like the tubes that lead up to the kidneys, right? They're both Y-shaped tubes that go up to some things. And I've seen this kind of thing messed up, before, because they're just like, oh, that's the urethra, or that's the oviduct, whichever. Look to what it's connected to, right? See where it's going and where it comes from. So understand the whole animal. All right, within the kidney. It's awfully noisy in here. All right, so here's a nice kidney model. There's some regions, obviously. Some of the parts are pretty simple as long as you've got the hard drive space to remember them all. There's just this outer capsule, right? What kind of tissue makes the outer capsule?
It's fibrous.
It's fibers. So in this case, it's dense irregular. So that's because the kidney is under a lot of pressure. It's like high fluid pressure in there, right? That's what's going on inside this. Wait, I skipped digestive system. That's okay, I'll go back. So this excretory system now has got, this kidney's got some regions, the cortex, the medulla, and then this part's all basically funnel, right? You can really see it on here, and students oftentimes will get confused between minor, major calyces and then the pelvis, right? Well, they kind of branch at the end. So this first funnel, it opens into a minor calyx. These two funnels together form a major one. When they get down into this region, it's the pelvis, right? So it's simply layered, and it's not too hard. And, you know, it's easy for me to say that, of course, but you know, it's just some parts. Blood vessels also, know those, right? Know those, that sequence of blood vessels going in and out. You might get one question on that, and again, big deal, one question. You missed 25 one questions and you're mid-C.
Where does the pelvis start?
Well, it'll be safe to say that the pointer will not be anywhere vague. So I can't tell you exactly where I would say it starts, but if this is a major calyx right here, maybe where two major calyces meet would be the pelvis. So once you've got the calyces starting to unite, then you've just got pelvis. So everything covered by my fingers is probably pelvis. Oh, he's okay. He's doing fine. What kids do. All right, so I went to the urinary system because that was next, and we went out of order in the actual the nephron. Right, so I'll stay on this order. So the nephron, right? Let's just refresh our memories here as to what it is. So this blood vessel right here, this is I call it a cortical radiate artery, but I think in the book it's interlobular, right? So the interlobular artery will have a branch that goes towards the Bowman's capsule. And so towards is afferent, right? If you have any knowledge of Spanish, the word means to, right? So it goes towards the Bowman's capsule. So the afferent or afferent, and then we've got this ball of capillaries. What's it called? Glomerulus. And then we've got an arteriole leaving. Efferent, right? Now most of the time, arterioles go into a capillary bed and leave as a venule, right? But we go arteriole, capillary bed, arteriole. And the reason is because we're going to go into some more capillary beds. So it goes arteriole, capillary bed, arteriole, capillary bed. These guys are called peritubular capillaries, or around the tubes. Now the whole process of filtration, reabsorption, and secretion is a little confusing to some people, but just think, where did every drop of your urine just come from, besides for your bladder? Where did it come? Where was it before it was urine? It was your blood, right? So all of your urine comes from your blood. It's made of liquid from blood. So this first process, shoves all that fluid into there, and that's called filtration. It only occurs there. The second two processes, reabsorption, and you want to think of that as, I'm going to take some back, right? Filtration is put it in the tube. Reabsorption is take it back. And we're going to reabsorb that stuff over the course of several places so that we can get a lot of that liquid back. So reabsorption is taking back this stuff. You're going to get back water and some salts and glucose and some urea even. And then secretion, it's not really at the end, but those two things of reabsorption and secretion go hand in hand, where you're trying to take back water and take back some of the things you need, and you're trying to shove out into this tube things you don't want, like urea, uric acid, some extra salts, that kind of stuff. So glomerulus, Bowman's capsule, this space in here, called what? Capsular space, which is within the actual Bowman's capsule, right? The capsule, the whole thing. What do you call the whole deal? A renal corpuscle. So how do I know where the cortex stops and the medulla picks up under a microscope?
When you see collecting.
You can see those, but you can see you can see tubes that may confuse you in the cortex and the medulla. You'll see those, if it's a good slide, you'll see those nice long parallel lines which are collecting ducts. But you'll never in the medulla see a renal corpuscle. All of the corpuscles, all the Bowman's capsules and all the glomeruli are in the cortex. Right? So if you see, when you see those guys cut off, and you see those those collecting duct lines, you know you're in the medulla. If you see any corpuscles, you're in the cortex. Right? What is the proximal convoluted tubule proximal to? The renal corpuscle, the glomerulus, or the Bowman's capsule, or whatever, right? So it's called proximal. Proximal means near, or close to. The proximal tubule is proximal to this thing. So even though in a model you may see it go squirrely all over the place, right? And you may say, well, this one's, you may not know where it is. But if you can follow it from here, if you can follow the tubule, this is all proximal convoluted tubule. After this loop, this nephron loop or loop of Henle, that's when we get to this guy, which is the distal convoluted tubule. So it's distal, it's farther down the tube. Right? Hopefully this is truly all review. Hopefully it's like not, what now? Let me get all that down. This is news to me. Kidney disorders, there's a list of 8 of them right there. You need to be able to recognize those, this is good, I'm glad I just saw this thing. This urinary test, right? Remember we did that urinalysis? We either used fake urine or real urine. My classes did some of each, right? We had some people on fake and some people on real. But just so you know, if you don't know how to read this, or if you don't remember, this is gonna be on it.
We didn't get to it in class.
Did you not? Okay. Well then I'll show you how to read it. So this is not review anymore. Now this is new. So there will probably be either one of these containers or a picture of this color pattern of this thing, right? So I'll stand in the middle. I'm gonna move. Sorry, I'm moving off camera. The assistant, the director of set 2 is gonna be on top. So these little pads right here have got some this little stick has some absorbent pads on it that have indicators saying if there's a certain molecule or pH or something on it, right? And so they line up just like this in a nice little row. So that when I hold them over there, they all line up with their respective row, which has on its left axis here a list of shorthand of chemicals. So it says SG. Okay, SG is a weird one, but SG is specific gravity, and it's the same. It's kind of the same thing that they measure beer with. They have like how many dissolved things are in it. So specific gravity simply tells you how concentrated your urine is. If it's over here, the bigger number means more concentrated. This way, it's more dilute. And you just try to match colors up. So this little pad up there should match with one of those colors, and you should be able to guesstimate where it is. And you'll dip this into some, or it will have been dipped into some fake urine. Then you got pH and leukocytes. What would a high reading of leukocytes indicate? Yeah, some sort of UTI, right? A urinary tract infection of some sort. What about, and then you've got nitrogen, you got protein. Any of these things can be indicative of kidney damage or some misfunction of your kidney, right? So that list of those, getting there, almost right there. That list of those homeostatic imbalances, so to speak, cover most of these guys. So there's one down there called bilirubin. Bilirubinuria. See that one at the bottom? All right? So bilirubin is a breakdown product of red blood cells. And if you've got too much of that bilirubin in there, that's indicative of a problem with your recycling your red blood cells. Called bilirubinuria. All right, well anyway. It's It's not hard to read if you just know how to read it. So, there probably will be a question of that on there. Did I leave the cap somewhere? Or did I bring? Got it. Okay. Digestive system. All right, what do we got inside of the rat or the pig? Let's start at the opening and walk our way through. Your mouth, right? Right? And there's even teeth. One semester I tied a little string around the front chompers of the rat and said, what type of teeth are those? What type of teeth are those? Incisors, right. So mouth and oral cavity and teeth, maybe I'm not going to go to I'm not going to look at a rat's back teeth and have you do, dentist work on them. What is the pharynx? It's your throat, right? Now I'm not gonna find the pharynx on a rat, even though it's starred. It's not gonna be there. You could see something like the esophagus. How do I know I'm looking at the esophagus rather than the trachea? The esophagus, first of all, lies behind it, right? So the trachea is this hard tube right here, ventral or anterior. Behind it is this soft tube called the esophagus. So that by location and by firmness, right? Trachea is cartilage ringed. Esophagus is soft. So you can see that. Then you get down to the stomach. That one should be obvious. The small intestine has a few parts, right? Small intestine's got the, here's the stomach. Yeah. It's got this bit right here, the duodenum. And then way down here at the end, it's got the rat's got a bigger cecum. So whatever, rat, cecum, like this, right? Mm-hmm. So this is duodenum and ileum. I don't know, if I'm paging through a rat's guts, I can't tell where the duodenum leaves off and the jejunum picks up. So you're probably not going to get a jejunum on there. But if you see a pin right by the stomach, and it's on the small intestine, that's the duodenum. If you see a pin right by the cecum, and it's on the small intestine, it's the ileum, right? So try and look for landmarks in the area. Don't just go, oh, it's a tangle of mess of guts. It may look like that at first, but it's not, I assure you. We'll try to set these things up so that if you know what you're looking at, you can tell what you're looking at. You'll be able to name what you're looking at. Okay, so those are most of them. Now there's some accessory organs in the digestive system, right? We've already seen one of them back in endocrine. Pancreas. So the pancreas works again. What are the name of the exocrine digestive system cells or tissues in the pancreas? Acini or acinar cells, right? And you can see those under a microscope, and I'll show you those when we get to that. Oh, it's okay. What is, and on a new rat, if I open up a brand new rat, what is the greater omentum? It's like a fat bag around the guts, right? What's the lesser omentum? It's above the stomach, and it kind of holds the stomach and the small intestine sort of up. You ever, when you got into your rat guts and you kind of opened them, when you pulled on these, let's say if this is the small intestine right here, and if you kind of pull them like this, you'll see this kind of a fan-like membrane inside of there. Can you picture that? Mm-hmm. That fan-like membrane is going to have blood vessels, and it's going to have lymphatic vessels, and it's going to have nerves. What's it called? It's called the mesentery, right? So those are pretty much the three connective tissue layers that you might have to name. And probably on the rat or the pig, you just get maybe mesentery and possibly greater omentum. It's hard to spot that lesser omentum in that sort of a setup. Hello, Dr. Gargan.
Hello.
What's up?
Just checking.
Okay. Maybe she's gonna quality check me. What's up, Hannah?
Can you describe the greater omentum again, please?
Sure. So I don't know if I can draw it, but yeah, I can. Sure. So the greater omentum, you got your small intestine winding its way around. The greater omentum kind of acts like a big pouch that covers this whole thing. And most of your rats once they've been dissected, once you've already gone in there and looked around for them, you've removed that stuff. So it would only be you'd only be able to really identify it as a intact structure in a brand new opened rat, which could very well happen, right? Could open it up.
I'm just counting.
Sorry, is that good, Hannah? Does that explain it? All right.
So we have to identify it on the brand.
It's on the, it's right there, so yes. So it's a possibility, right? The lesser omentum's, in all honesty, harder to spot on a little animal like that and have it really easily identifiable. But on a model, you can see it, or on a diagram, you can see it.
So would be the mesentery.
The mesentery is this thing that kind of holds the small intestine in place. It's a big fan of shiny sort of membrane with blood vessels in it that extends out from the dorsal body wall and kind of keeps those organs and supports them. It supplies and supports the small intestine mostly, and the large intestine. All right, on a model, which I'm not going to go digging around for, but on those models we saw, they're a bit more obvious what the parts are, so you should be able to see the ascending, transverse, descending, sigmoid, colon, rectum, right? So those parts you need to be able to identify and know a or two main functions of all these organs, right? Don't mail it in and just say digestion, right? Don't do that. If you want to say chemical digestion or absorption, right? That would be a good thing for the small intestine. If you want to say, yeah, well, vitamins are going to be absorbed most places, but the large intestine has vitamin absorption, right, from the bacteria. So you could say that for that. Water reabsorption, concentration of feces. So try and identify a specific role for each of those things. You don't have to, you don't have to go through a medical dictionary and find every single thing it does. Just a couple of the more obvious ones. And again, I'll get to the slide stuff here in a bit. The secretions of and these are in your book on page 120, and they're listed by region. So you can see, and this again is not a complete exhaustive list of everything that your intestines and your stomach and your liver and all that make. But it is some of them, right? And like for instance, bile, that's the only thing you gotta worry about out of the liver and the gallbladder, right? You know what it does. Don't say it has enzymes in it, right? Know what it does, it's got a simple job. Also know that the gallbladder doesn't store it, doesn't make it. Gallbladder just holds on to it. And some of the enzymes actually have names that tell you what they do, right? Lipase, protease. Yeah. Now amylase. Yeah. Okay, we skip nutrition, so don't bother. Yeah, it's kind of cool. You got it in lecture. All right, now we go on to reproductive system. So now we're getting into recent history. Now we're into our recent memory. What do we got inside of a rat? We got two different sexes. So we've got in a rat, let's say with a male, what kind of things can we spot on a male rat or pig? Testes, okay, and roll, right? Sperm manufacture, testosterone production. Well, I'll show you the slide and we'll see the different regions, right? The bag that holds the testes, scrotum, right? Now, on my reproductive system quiz, I pinned the epididymis of the testes, right? And I had a lot of students just say testes because they didn't take their time. They knew what the epididymis was, but they didn't take their time to look and see that it was a two-part structure and that it was very clearly in the epididymis. So don't just jump the gun on it. Oh, I know that one, testes, right? I had one student answered scrotum. I don't know why, but it was not that. So don't be hasty. You got time. And what else? So testes. What else we got? So testes, epididymis. Yeah, the ductus or vas deferens, right? So just follow the sequence, right? Sperm are made here, they mature in the epididymis. They move up the vasa deferens, the vasa deferentia during ejaculation, right? There's some glands which you can see on the rat. You can definitely see the seminal vesicles, right? You can definitely see those.
You cannot see the bulb one.
I'm not going to find that.
Yeah.
Prostate, you can see that. If it's underneath the bladder, you see a wad of tissue around the urethra.
What is it?
Prostate.
About the rat itself or just the model?
Pardon? Definitely models, but theoretically, you can see it on the rat. Let me make sure. If it's got a star next to it. Yep, star. Seminal vesicles, prostate glands, not bulbal urethral gland. Okay, female. Start at the beginning again. Again, rat or pig, right? Ovaries. So now, did you guys, we had a pregnant rat? Yeah, we've got a pregnant rat, so we can still see it. We have a pregnant rat here. If you didn't see it, before we leave today, we can check it out, okay? Did you take out that little baby rat?
Yeah.
That placenta was awesome. That was really cool. There was the baby rat with like a umbilical cord and a placenta. It looked like a model, because that placenta was like a little.
You found out? Yeah.
It's probably in their hand. I don't know in their bag, but we'll get it out. I'm sure it's in there, unless somebody threw it away, which I hope they would. So ovaries and what occurs there, right? Microscopically, a little bit more challenging. The models, kind of a representation of what you can see microscopically, but again, it's made out of plastic. What's after the ovaries? What are these little grippy dealies? The fimbriae are those finger-like extensions, right? And then this is the infundibulum, and then you've got some other parts. But this whole tube right here, I'll take different names for it, but it's listed as fallopian tube. But I accept uterine tube, uterine horns, oviduct. Yeah, my term is oviduct. That's the one I tend to use. But in this pregnant rat, you can see why it's really a uterine tube, is because it acts as a uterus. Then you get to the uterus, right? What's its job? Yeah. So yeah, development of the embryo and fetus, implantation, that kind of thing, right? There's layers in there that you have to be aware of, and their roles. They're yeah, the endometrium is the inside layer. What's it good at?
It has two layers, base layer and functional layer, which is for okay.
You're doing all right. No, that's good. But yeah, so generally it's a highly vascularized layer that's going to interface with the embryo's tissues and form that composite tissue called a placenta, right? So then we get to the bottleneck at the bottom of the uterus, cervix, right? And that's literally acts as a bottleneck. Okay, those embryos those fetuses have to get bigger and bigger and bigger, and they kind of need that's their room until it's time to give birth. And then the exit is the vagina, right? That's an internal structure, and its main purpose is not. This is one thing I stress to my class. The main purpose is not a birth canal, but it's where the sperm go, right? Because we conduct internal fertilization. All right, now when we get down to the actual cells, the actual reproductive cells, the sperm cells have some identifiable parts, right? There's like the head. What's that little enzymatic tip on the sperm cell called? All right, so that's like a little enzymatic tip that's going to act as a thing that's going to dissolve its way into that oocyte. What cell does the sperm cell run into in the oviduct? What's the name of the cell that it fertilizes, or that it bumps into first? Called a secondary oocyte. Right? It's called an ovum. This is part of that development thing, but right after that sperm cell enters, the egg cell that oocyte undergoes meiosis II. So it becomes an ovum after, in the process of fertilization, depending on what you want to.
So before the ovum is the secondary oocyte.
Right. It's the secondary oocyte before it comes an ovum, right? It's already haploid, but it goes through that second phase right after that sperm cell meets up with it. And I'll give a brief sketch of meiosis here. What's the difference between spermatogenesis and spermiogenesis?
Spermatogenesis forms the
Spermatogenesis makes that spermatid, right? And the spermatozoa is kind of the finished product that you get in the epididymis, right? So it's like kind of like dealer prep. You add like a undercoat and some, like you polish the tires, give it a long tail. And again, we've got, at the end of this, we've got a hormone section. You thought you were done with hormones when we got out of endocrine system, but here they are again. Source and function, these are 2 columns on page 130 that make perfect sense. Again, if you can know, in some cases, like say for testosterone, right? Testosterone, you could say testes, I guess, but specifically, if you look at it under a slide, if you look at it underneath the microscope, it's the interstitial cells. It's those cells that are in between those seminiferous tubules. And prolactin and oxytocin, right? Where are they made? You remember? Pituitary is for sure, but prolactin is anterior and oxytocin, hypothalamus made, stored in the posterior, right? And they have slightly different jobs. Prolactin, it's very pro-milk, right? It loves milk, it makes milk. Oxytocin does a couple things. Uterine contraction and milk ejection. All right? Gotcha. Good job, you guys. All right, development. Development. So here we are. This is kind of a continuation of the reproduction section, obviously, right? But this is reproduction in action rather than just the system. So at this point now we're talking about once you've got that reproductive ball rolling and you are in the process of making an embryo and developing it and supporting it and so on. These erasers, I don't know if it's the pen or the eraser or the board. A lot of different options. Where do I want to start? How do you guys feel about those stages of embryonic development? You don't like it? You feel uncertain? Anna? Anybody else? So, all right. So here is Anne. Pen's even worse. Be gone, pen. Wrong basket. Huh.
I had a red one.
Right basket. Aha, there we go. So this is a secondary oocyte. And here comes a sperm cell, right? What is the ploidy of these two cells? Ploidy means how many chromosomes they have, right? So in humans it's 23 and 23. So, which is half of the number that we have in our normal cells, right? So haploid. It is N also, correct. So if 46 is symbolized as 2N, then 23 equals 2N divided by 2, which equals N, right? So they call this haploid, and they call this diploid. So when we produce gametes, what's a gamete? Sperm cells and egg cells, right? That's just a term, the generic term for both sperm cells and or egg cells or oocytes. So when we produce sperm cells and egg cells, we produce it through a division called meiosis or meiosis, right? It's meiosis, it's not urosis. So meiosis divides the number of chromosomes. We go from diploid to haploid. So that way when these two meet up, when these two crazy kids meet up here in that dark alley, it is a dark alley in here. No light. Then you go back. This is not shocking to anybody except for not my students. My students are like, yeah, day in the life. Everybody else is like, oh my God, this guy's insane. So then you go back to, then you return back to your normal number of chromosomes. That's the number of chromosomes that we have. That's how we make another person, right? Chop them in half. Somebody else chops theirs in half. Put them back together. What is me putting my chromosomes along and putting them together with some other person's chromosomes called? Crossing over is a process during my making my own sperm cell. But what's the overall process called? I'm taking genes from one animal and putting them with genes of another animal.
Gene recombination?
There you go, gene recombination. So genetic recombination is any mix-up or recombination of the genes. So genetic recombination, oh, I'm just kind of jumping
This is
around.
29.
I'm just jumping around.
Yes.
Is that okay?
Okay.
I'm just kind of following my line of thought here. Because I think
Follow
I'm,
your heart.
huh?
Follow your heart.
Yes, I will. Straight on, turn left at the second star and straight on the morning. So, but this is because you got to look at it as a whole process, right? My reproductive organs are there to make these cells, which are there to recombine with somebody else's cells in a number of different ways, which include that crossing over bit, which occurs when? The prophase 1, right, of meiosis. We had segregation of chromosomes and independent assortment of chromosomes. And this just means that the chromosomes don't have to go in any predictable way. So I can have red and blue, black and blue like that, or I can have it go like this. So they kind of mix up. When I'm conducting meiosis, I don't make sperm cells. My sperm, like hundreds of millions of sperm cells, don't all have either all of the left. It's not like left twigs and right twigs. It's not like I've got all the left chromosomes in one sperm cell and all the right chromosomes in another sperm cell. Any one of those 23 pairs can go in either direction, which is a really good way to mix up your genes. It's a really good way to kind of have you're a gambler. Things that do sexual reproduction are gamblers. We're making a bunch of different type of organism. We're not trying to stick with this conservative, straightforward, this worked for me, so it's going to work for you type of thing. We're living in a changing world, and we've got to mix up our genes and hope for the best, right? Some do better than others. That's right. I know it's a super gene. Random fertilization, right? In this case, this guy won, like in this example, right? But there was a whole bunch of, also-rans that could have been the one. This oocyte happens to be the one that's up to bat this time, right? But not every oocyte gets fertilized, and not every fertilized egg gets, is successful. So, which one of these oocytes is going to win the lottery and become a baby? It's random. And all of these means of mixing this stuff up really add to our genetic diversity a lot, as sexually reproducing organisms, as humans in this case. Well, in any case, what's this single-celled fertilized egg called? That's got to be in there, right? A zygote, right? So a zygote is just, once that sperm cell fertilizes that egg cell, boom, you've got a single-celled diploid thing called a zygote. Now the next few stages, hopefully nobody misses. 2-cell, 4-cell, not 3, they double, right? It's like computer memory. 8-cell. And I'm not gonna, I don't know if your teacher's gonna give you 16 cells, but I'm gonna stop there. I don't want to have somebody going 1, 2, 3, 4, 5, 4, 1, all those counts. You can count 8. But now this 8-celled stage, doubles again. Pretty soon you've got a solid ball of cells. And one thing you'll notice is that this solid ball of cells isn't much bigger, if any bigger, than the original cell. And that's because these cells have been getting smaller and smaller and smaller. What are these early stages of division called? Blastomeres are the names of these cell. This is a blastomere. Blastomere is the early cells that are products of, what kind of division is this? It's called cleavage, but what's the is it mitosis or meiosis? Mitosis, right? Meiosis makes these guys. Mitosis then makes more of these haploidiploid cells. So blastomeres, the names of these guys, these divisions, these early mitotic divisions are called cleavage. Because of the way it looks under a microscope, it looks like they're being chopped in half by an invisible axe, sort of. Once you get to a solid ball of cells, what's it called? Yeah, morula or morula. It's probably actually morula, not even my Latin. So it's, well, whatever. This is a solid ball, so if I cut it in half, it would be cells all the way stuffed all the way through. But at this point, the embryo starts to develop a little bit of complexity, and it's gonna move all of those cells to the outside, and I've shorthanded the cells there, forming a hollow ball called a blast. If it's the starfish, it's a blastula. Okay. If it's in an organism like us that is got internal development of the embryo, and you've got a placenta, and you need to make tissues that are going to interface with the mother, it's called a blastocyst. And a blastocyst you can identify by the presence of these cells that kind of accumulate at one pole. So a blastocyst is this one, and that is your inner cell mass. And the inner cell mass becomes the embryo. The inner cell mass becomes the embryo. Now, in this blastocyst, what's this part called? The outer ball. The whole thing's called a blastocyst. Right. So that this thing's called the trophoblast. Now, it's harder to draw and really visualize on a blastocyst, but the same process is the same. This guy's going to start folding up. This embryo is going to start folding and making more layers and becoming more complicated. In a starfish, that's going to look like this, right? Where you've got an indentation, with that indentation eventually becoming more complicated like this. And this guy right here, this stage where you've got that dent and those folds on the inside, what's it called? These are called, the process is gastrulation, but each of these guys are called gastrula. So the gastrula is the stage where you actually see that folding occur.
Is that stage where the germ
Is that the stage of the what?
germ cells appear like germ
The germ layers? Yeah, this is the stage where you're going to start to see the initial dedication of those cells to becoming certain types of cell. So prior to this, these are just cells, just like kind of stem cells, right? But as you get further into this development, you're going to have, roughly, this is ectoderm, this in here is endoderm, and then you're going to get patches of cells forming in here, which is going to be mesoderm. And those cells have different destinies, right? They become different stuff, and that's, kind of have a vague idea of what that is, right? Ectoderm, mostly epithelial stuff in mouth linings and central nervous system. Endoderm, organ linings. Mesoderm, most stuff. Mesoderm's most of your muscle, your connective tissue, your blood, your bone.
Gastrulation is a whole process.
Yeah, gastrulation is the process of that blastula becoming a gastrula. Ooh, I got a crick in my neck. I really have to stretch it out. Hold on. It's really tightening up on me. Oh, all right. All right, I'm going to try to move it along here. Fetal circulatory system. All right, heredity. I'm going to be brief on this. I'm not going to go past. I'll go to 5:30. But After that, I'm off the clock. Let me see what time the next lab is. What day is today? Yeah, cool. There's more room than we need. All right, I'm going to give you guys, you're going to get a question or two or maybe three on this whole heredity thing, which is hard to explain in a single 2-hour lab, especially if you don't explain it very well. So hopefully I'll give you a kind of a brief breakdown. So do you remember in meiosis, we go from diploid to haploid, right? We go from 46 to 23. In this case, I'm going to go from going. Let's just say in our case, we've got an organism that doesn't have 46 chromosomes or 23 pairs. It's got 2 chromosomes, 1 pair. But it's the same process. If I just drew 22 more pairs of chromosomes, it would be the same idea, right? But we've got two chromosomes. These guys are called homologous chromosomes, or homologs, because they have the same genes on each as the other. So this one, I could draw a little hash mark there to represent a gene. And that little hash mark now could be the gene for I don't know.
Blue eye.
Thanks, but yeah, I'll do eye color. We'll do eye color, even though eye color isn't a gene that just has, isn't a trait that just has one gene. But let's just imagine, let's say tooth sharpness, because that's something made up that we can't necessarily connect. So this is going to be, we all have teeth that have relative sharpness, right? We've all got teeth. Some of us have dull teeth and some of us have sharp teeth. Let's just put it that way. So those different, they both count as the gene for tooth sharpness. But there's 2 different versions. There's a dull version and a sharp version, right? So what happens is that oftentimes one of those alleles, one of those versions of a gene, which is what an allele is, one of those alleles will express itself, I'll stay in the camera, one of those genes will express itself preferentially over the other. So if they're both present, one's going to be expressed and one's not, right? So if you have, you know how you have I've got 2 friends, one's really loud, one's really quiet, okay? If they're both in a room together, what does the room sound like? Loud, because even though the quiet one's being quiet, the loud one's expression is dominating that quiet one. The only way for the room to be quiet is to have the quiet one be alone or to have 2 quiet ones, right? So with tooth sharpness, let's just do loud and quiet. I'll switch it to loud and quiet. So in this case, gene A, or little a, represents loudness, right? So we'll use the big A as dominant because this is loud. When loud is expressed, there's loudness. This one is quiet. So if you want to be quiet, if you want this to be a quiet person, you've got to have 2 of these things, right? So right here we've got this guy. So this person has 1 copy of a loud allele and 1 copy of a quiet allele. That is their genotype, which is the actual combination of alleles. So whatever those genes are, whatever those letters are, is that organism's genotype. Is this person loud or quiet? Loud, right? Because it's got this one, which is dominant to that one. So the phenotype is loud. So yeah, it's kind of the trait, the observable trait. Now, if we want to do a cross, when these 2 people meet, right? Here's a loud person, and I'm going to put another X here, but this just is the genetic cross, so they mate, right? That's what that indicates. So a loud person finds true love in another loud person. Incidentally, when you have 2 different versions of the gene, there's a big one, a small one, or whatever, it's called heterozygous, right? That's the description of that combination. If they're the same, they're homozygous. If they're different, they're heterozygous. Now, these guys undergo meiosis, and that whole meiosis process, which I'm not going to have time to get into, that whole meiosis process involves meiosis 1, which has prophase 1, metaphase 1, anaphase 1, telophase 1, 2 cells that are haploid, right? Then those 2 cells go prophase 2, metaphase 2, anaphase 2, telophase 2, and you got 4 cells now, right? So these individuals are going to produce gametes that have one or the other allele in it, but not both. So when this, we'll say this is the guy, they can either produce a sperm cell that's got the allowed allele or the quiet allele, not both. The female, she's only going to produce one egg, but for mathematical purposes, it could be either one. It could be a loud egg or a soft or a quiet egg, right? Now this is how you make a Punnett. This is when you make a Punnett square to determine what kind of offspring can come of it. This is a really simple type of genetic cross that you can put on paper. So what I do is I just have, there's 2 options over here. There's 2 options over here. That equals 4. So I just need to make a 4 box Punnett square. And I put the letters along each axis, and then I just run those letters across. And I'm going to keep them all the same just so I can keep track of them. I'm going to put the big A's first and the little a's second just so I can keep track of what I'm doing. But this is what you'd call, this is the possible offspring genotypes. And genotypes are the combinations of those alleles. So this doesn't mean that these 2 people are going to have 4 offspring at once. That's not the case, right? But these 2 people are going to have 1 offspring, and it's got a chance of being any one of these things. So the odds of it becoming a big A, of these 2 people giving birth to a allowed homozygote are 25% or 1 out of 4, right? The odds of them producing a loud heterozygote, 50% or 2 out of 4. And the odds of them producing a quiet, obviously a homozygote, 1 out of 4 again, or 25%. So if you said, if I asked you what's the offspring, the expected offspring, expected genotypic ratio of the offspring, I'd say 1 big A to 2 big A to 1 little a. That's the genotypic ratio. What's the phenotypic ratio? What are the phenotypes here? What's the traits? The phenotype's the actual trait. Loud, right? Quiet. So 3 loud to 1 quiet. And that's the phenotypic ratio. So you're going to have to be able to do that. Now it works the same way with sex-linked traits. Sex-linked traits are function the same way. It's just that they're on the last set of chromosomes, which also determines what sex you are. So males have a different chance of getting sex-linked conditions or traits than females do because they've only got 1 of each chromosome, whereas females got 2 of those. If there's a gene that's on the Y chromosome, what chances do females have of getting that gene? Nothing, right? 0. So if there's a trait or disorder that you find on the Y chromosome, females don't get it. If there's a trait or disorder that's on the X chromosome, males only have 1 chance to get it, one X chromosome. Females have 2. Wouldn't that seem like it would make females more likely if they've got 2 chances to get the trait or the gene or the allele, that they'd have twice the odds of getting a disorder or something as a male?
But no.
Wait, no. But yes, if it's a dominant trait, if it's a dominant allele. But if it's a recessive allele, then a female and a male right here. So there's a female and a male. If it's something like hemophilia or color blindness like we had in lab, we talked about in lab. So color blindness, normal vision will code for big A because that's the dominant trait. Color blindness, little a, the recessive trait. How do you exhibit a recessive trait? How do I exhibit quietness? How do I get the phenotype of quiet? That's usually how people say it, right? The thing that, but it's kind of missing the point. It's not to have two of them. It's just not. It's the way you get that recessive trait is to not have any of the dominant trait. So it's not the fact that there's two little ones. It's just the fact that there's no big ones, right? So
is it the absence of the dominant?
the absence of the dominant trait lets the little one, that's the recessive trait become expressed. So in this situation, males only have one shot to get the recessive allele, but if they get it, they've got it, right? So if you draw the short straw here and you get this little A, well, you're colorblind. Because all you need is one copy. What are the chances of females getting the copy? It's reduced, right? Because now you have to get unlucky twice. Now you have to get a little a and a little a, which is much less likely than getting a little a and a big A, especially when they're rare, which they usually are.
So so that's.
No,
Yes.
no, it's both fault. If the boy could get, if I was colorblind, it's my mom's fault. If I'm colorblind and my dad's colorblind and my mom's not, it's still my mom's.
Yeah.
Because I'm a boy, right? Which means I got a Y, right?
Uh-huh.
And where do I get the Y? That'd be a neat trick. But you get it from my dad, right? I got it from the guy with the Y. So he gave me the Y. If I got a little A, I had to get it from mom.
Uh-huh.
How could I get it from mom if she's not colorblind?
It's yes,
Because she looks like this.
yes.
Okay. That's about as crash of a course of that as I can do. But let me look at some slides real quick. Okay. Let's give you some slides that I have seen in the past. That I'll give you some of the more problematic ones. Is that a word? Problematic. I've been in this room 1,000 times. So like, where's the books? Now if the microscope just works, I'll be okay. Oh, and if anybody has to go, they're welcome. Don't worry about being rude. If you gotta go, you gotta go. If you want to stay, you can. Now, the thing about slides, and my students have heard this a dozen times, is that if you put them in there yourself, you've looked at the label. So you know what you're looking at. Oh, look at this. We got custom made, what's that? Oh, you don't see it yet. Panasonic sign. Somebody already had a nice slide in here, so I'll show it. And the crowd says, survey says, what is that? It is a gastrula. So see that little dent right there, right? That's the key. You can probably find there. That would be a blastula, and then you see up here. Ooh, you see up here are two gastrulae and a blastula. At least that's what I call it. So this is one of those slides that has a mix of advanced or more advanced and less advanced ones. Chances are, if you get a gastrula, it's going to look like a classic gastrula. It's probably not going to be one of those bullseye looking ones. At least I hope. 36. Okay. But let's start a little farther back. That was just a target of opportunity. All right, as for the some of the tissue types, you still have to be, you're still responsible for knowing tissue types. but, you know, the fewer of them than you did in ANP1, and you're better at it, I hope. So what is that? It's the esophagus, how do you know? Stratified squamous epithelium, right? That's what this stuff is. And this is something that you get, for the people that said esophagus, this gets so that you automatically can tell what that is. And this takes, you know, practice. How do you get to Carnegie Hall? Practice, practice, practice. A joke from the 1900s. So now what I've done is I've found a region here that is a clear transition between that sort of tissue, this stratified squamous, and then this sort of tissue. What's connected to the esophagus? The stomach, at least on this end. We don't have any mouth slides, right? So you know it has to be the stomach. And then knowing what you know, what type, you don't even have to look at it, what type of epithelium lines this? Simple columnar epithelium. And we could probably go in there and, you know, if I wanted to give you guys a slide like this, that would be enough to identify these things. And if I had a pointer right here, I said name the organ, you'd say esophagus. If I said name the tissue type, you could say stratified squamous. Name the organ, stomach. Ooh, what are these? Yep, they're gland. They're not goblet cells. They're mucosal glands. See them? See how that's like those are like glands that are in gastric pits. So they're going to be glands that are going to produce a number of things, but mucus is one of them, and acid is another one. Let's see if we can identify the simple columnar epithelium. Now, when we get to the stomach and we get to this tissues, it's probably not going to be too much of this up close stuff. But if I wanted to ask you about an epithelium type, I mean, I could do it right there. That looks not unreasonable. I slide my slider. Slider won't slide. This newfangledy machine. Whoa, there we go. Yeah, those cells look simple columnar to me. They're all one length of a cell. You guys can identify the ileum, I hope. Ileum's pretty easy, I think. How do we know it? Peyer's patches. Where is the ileum? Number 27. And just as luck would have it, there's one. Let's see if this works. So those guys right there, right? Those things are these things right here are Peyer's patches. And they're anytime you see this circular sort of ring here. If you see that ring shape, you know you're in a tube. And if you see this big lumen in the middle that's all broken up like this, you're probably in the digestive system. We're not doing cardiovascular system, so you're not going to get any major veins or blood vessels here. And doesn't look anything like a trachea. What would a trachea have out here? Yeah, hyaline cartilage. They're all going to have smooth muscle, but the gut is going to have a lot more. And then this stuff, these Peyer's patches are right here. Again, simple columnar epithelium out there in the epithelial along the lumen. How do you tell the duodenum? Brunner's glands or duodenal glands, right? Let's find that. He's being a patient little guy pretty much, really. And there they are, right there. They look really good, right? So now this looks clearly similar in some regard to that ileum. You've got a mucosal lining with the what you can infer to be simple columnar epithelium. You've got this stuff out here. What tissue is that? Muscularis externa is the layer name. What's the tissue type? Smooth muscle. Again, that's one of those that, try to remember, if it says name the layer or tunic, you could say muscularis externa. If it says what tissue type are we talking about, like this is the mucosa, tissue type is simple columnar epithelium, right? But this layer, found in the same place as those Peyer's patches, is completely different, right? These duodenal glands are produce digestive enzymes and stuff. They're way at the top of that small intestine. The stuff at the bottom, the Peyer's patches, serve a completely separate purpose, which is like malt. It's like a lymphoid tissue, so it's like infection protection. And the jejunum is just generic. It's generic. It has nothing really going on. So if you don't see duodenal glands or Brunner's glands and you think you're in the small intestine, go with duodenal glands or Peyer's patches, go with the jejunum. Let's look at that kidney. I promised you that there was no corpuscles in the medulla. So here we are. This is, and you can actually see some stuff. I just told my lecture class this, but I said the outside of the kidney is a renal capsule surrounded by perirenal fat. So this is that fat tissue. Can you guys see that?
Yeah.
That looks like adipose tissue to you, I hope. It always looks the same to me. So this is adipose perirenal fat, the renal capsule, which is dense irregular connective tissue, and then we go into the cortex. So we see lots and lots of these little structures here. Now don't get these confused with the pancreatic islets. Don't do it. They're different. Notice the gap. There is a space between this stuff and the surrounding tissue because there is a space between this stuff and the surrounding tissue, and that is the capsular space. So we're looking at a glomerulus right here. The pointer is on a glomerulus. The pointer tip is in the capsular space. And if I zoomed in really close and turned off lights, I could see the cells that line it are simple squamous epithelium. And then there's a bunch of cells glommed on there called podocytes. But there's a space between this stuff and this stuff in the kidney, which is going to be a dead giveaway to tell that it's not the pancreas, right? In the pancreas, which I'm not going to, well, just to show you the difference. I'm not sure if you want to see this, but somebody must.
Is the pancreas on the
It's fair game because it's part of the digestive system. It wasn't in that slide box, but it is a. Let me before I leave this, let me finish it up. So here we are in the cortex. We move farther deeper, I guess. And we still see lots of these corpuscles, but then somebody said you get to see these collecting dots. So down in this area, this is medulla, this lighter colored area. It's not always going to be noticeably lighter, but do notice that there's no corpuscles in it. Not a single glomerulus or Bowman's capsule to be found. So that indicates, and you can see kind of a demarcation there. But another thing you might want to notice is that there's lots of nephrons up here, lots of glomeruluses and Bowman's capsules up here, right? In the extremity of that cortex. But if you get closer to the middle, you've got fewer and fewer, right? Here's one really close. what classification of nephron would that glomerulus belong to? Juxtamedullary, right? So don't get juxtaglomerular and juxtamedullary mixed up. Juxtaglomerular is a dead apparatus, but I don't think you have to know in lab. Juxtamedullary is a positional term because it's next to the medulla. What's the job of these guys? Concentrate urine, right? Produce urine with or reduce blood, volume loss. And these guys are just your everyday run-of-the-mill urine makers.
Does it also sample
Blood
blood and
and food.
fluid?
That's the juxtaglomerular apparatus, which is you only have to deal with that in lecture. Okay, so you don't have to deal with that in lab. It's not on the list, so I'm not going to show you. Then we get down to here, and we've got this is some internal structures that I can't tell you what they are, because you don't have security clearance. No, but, because I don't know. This is like the pelvic region or the calyx, I can't tell. Here's some blood vessels, don't know what they are. Don't know, don't care. Turkey. Where did we just get there? All right, so let's go back to this pancreas here just to show you the difference. Hopefully your memories are still fresh over there.
Like email? Yeah, I know. I'll try.
So first of all, the structure's not layered the same. There's no big fibrous capsule on the outside. There's some fat. But if you got to look at this guy right here, and you weren't, on the ball, you might see how this could be mixed up with a kidney. But no capsule and little to no space between these tissues. Really, it's only an artifact of the preparation. And they're also real irregular, right? These islets of Langerhans are irregularly shaped. They're not all roundish. Most of those glomeruli were round. So kind of qualitatively, there's a significant difference between them. Everybody cool with that? Fair enough? These cells out here, by the way, not these. These are the exocrine portions. Endocrine portions. The exocrine portions are these things, and that's those acinar cells or acini. And they produce pancreatic juice. Where does that pancreatic juice go? How does it get there? Pancreatic duct, and then the pancreohepatic papilla, right? Which empties into the duodenum. Down in front. Just kidding. How about this? We'll take. I got 7 minutes. We'll take audience requests on slides preferably, but if you want to see something else, let me know. But that means you got to tell me something.
Oh, the tube.
The tube.
The tube.
Oh, the a tube. Oh, the tube slide.
Like what we need to know on that.
Okay, on the slide itself.
Yeah, number 22.
All right, I'll find it.
Can we also see
The what?
Is it possible to explain the difference between salivary gland and the lungs, because they're so similar?
Oh, salivary gland and lung?
Yeah.
All right, I'll show you that. I'll show you that. All right, so here's a tooth. It's on the lowest possible magnification. This one's a little lower, but why? So this, now the tooth, ooh, jeez, I'm going off of that. That lens sucks. All right, so here's a tooth. Now, it's kind of a weird thing to put on a slide because it's so big, right? It's like there's a lot of stuff where you wouldn't be able to necessarily tell what this is. I wouldn't put this on there like this. So for the tooth, if it came on a slide, which I've seen it on there before, you'd probably get something like this at least, where you'd get the obvious outside point, and you got this sort of monolithic appearance here. It's just one continuous thing, right? Where you'd have the outer layer being the enamel, and then the inner layer being the dentin. You can see a margin there. And then deep in here, this is the pulp or the pulp cavity. So on the tooth slide, that's probably it. I mean, I can't speak to exactly what your teacher's gonna give you, but if you could recognize, if I could get that dentin on there, or that pulp on there, like this, I guess theoretically I could say, now we can see pulp, dentin, enamel, and then this edge, this is the gums, right? This is the gums, so gingiva, or the edge of the gums where they meet the tooth is called the alveolar margin. So that's about it. That's the most you could be asked, and I'd guess they'd keep it to something, just one of these 3 parts. 1, 2, 3 parts. You won't have to determine if that's an incisor or anything from that, even though it looks like an incisor. Man, my neck is killing me. What am I showing you? Lung and salivary gland. All right. So let's look at salivary gland here first. Now these glands, there's some slides that have multiple different glands on them. They'll have the parotid and the lingual and the submandibular. This one's just lingual. And it's not a great one because it's mostly, this one's really compact. So let me see if I can't get a better one. Let me see if I can't get one that looks more like a typical one. So 21, huh? Here's the salivary gland composite slide. So this is, I think this is more like the slide that you guys looked at. This one right here? Okay. Oh, zoom in? All right. Let me just look around. So one thing you want to notice that these guys all have in common are these sort of clusters of cells with some tubes in between. And now this, trust me, this looks different than, this looks way different than a lung.
Yeah, it is.
This one, you can see clusters of cells with tubes again. And then up on this one, which is probably the one that you get most confused. Ooh. I went really off board there. This one. But, I mean, I don't know, it's probably just, I've looked at them a bunch, but this slide, I don't like this lens. Let me go back to this. So, now that would be confusing, right? But that's not what it's going to look like. It's going to look like this, right? And you can see that even though these initially appear to be hollow, right? This is going to be, again, acinar tissue or acini that are secretory cell, patches of secretory cells. So and this is why you got to get good at playing with these toys here, right? I can turn that thing from looking like a lung. Now I'm not changing the light. I'm just changing that diaphragm. I changed it from looking like a lung to looking like not a lung, right? Lung, not a lung. And even then, it doesn't really look like a lung to me. So you've got to get good at that. Don't forget those tools on that microscope are there for you to play with, right? The light intensity knob, diaphragm lever thing, how close you want to put that condenser to the screen. But salivary gland is salivary gland. I don't think your teacher's gonna, I'm pretty sure they're not going to ask you which salivary gland it is, right? Is it parotid or whatever? No, that seems like out of bounds. I know me and Dr. Gargan are not going to. And I would guess nobody else is. So I'm gonna compare that to the lung, which is number 19. And we'll see what we can do. This is still the same level of magnification as that salivary gland was on. And if I go back to this power, and I adjust the different settings here, you can see now that when it's properly focused, that it doesn't look, they don't look the same. At least I don't think they do. You've got a lot more open space that's literally airspace in this lung, where in the salivary gland it was all packed in, except for when you bleached it out. Now in here, there's a couple things you might have to recognize, and you probably won't get to be too. They probably won't be too much of a stickler between what counts as a alveolus or an alveolar sac or an alveolar duct, right? I can tell you is what I'm going to do, and that's nothing. But if I go to any one of these regions and I zoomed into this, like so, this is a pretty classic look, pretty typical look at the alveoli. So these are alveoli. These are the sacs that you breathe into to exchange gases. I thought I saw there's a blood vessel right here. This is a blood vessel. It's like an artery, but I wouldn't bet my life on. And I know I saw a bronchiole somewhere, right there. So this is a bronchiole, and one way you can tell is that there's no, big smooth muscle layer, right? There's some, and this is a fairly large bronchiole. I don't see any hyaline cartilage, mostly smooth muscle and dense irregular connective tissue lining. But then again, then when you get down to the surface here, this is simple squamous epithelium. Same thing as you get in a blood vessel, but much more irregular border. So bronchiole. That's all. Show you what? Too obvious? Jejunum? Yeah, I'll do that, and that'll be the last thing, because I'm honestly virgin up here. I need a, I'm not asking, but I could use massage. I'm literally not asking. He asked us for a massage at the end. What's that?
Extra flex.
Yeah, am I your teacher?
Yes.
Then no. That's even weirder. All right, 26, where are you? The human jejunum. I'm trying to find one that is a circular, like a tube, the tube-shaped version of it. I'm getting a lot of cross sections here. Going. Okay, I don't have any on me that would be circular, that would be able to be compared to the other one. If you guys can find one over there, I need one that's like round, so it would be apples to apples. Slide number 26. Is that round?
No.
You want round? Yes. Here's a round, that's not round. That's like a sagittal section. You got one? Yeah, this will work. Don't put your box around me and give it back. So just for sake of comparing apples to apples, let's look at this one, which is a brownish one. So there's still a submucosal layer. This one's kind of bleached out, but we still have this this is a classic sort of smooth muscle of the gut out here. And then we see the lumen, right, the mucosa in here. But then the stuff in between is neither fish nor fowl, right? you don't see those really tightly packed duodenal glands, nor do you see those big, darker patches of Peyer's patches. So this is when I said it's kind of generic gut. It's just a long tube that you do a lot of chemical digestion and chemical absorption.
Which one's this one?
This is the jejunum. And this is a pretty light stained one. You still have some mucosal glands right here, right? But they're not, the duodenal glands are located in the submucosa, so are the Peyer's patches. And so in this guy, you just got, and actually this is probably an artifact, this is probably more tightly packed in a intact specimen. This is a really bad one. Yeah, it's not terrible. But just kind of some generic connective tissue, no glands to speak of. All right. Okay. Well, that's all I got. So, if you, oh, here, let me get you back to the slide.